Okay, the topic of this project is quite different from everything we heard today, but there are some aspects of it that can be useful for people around here, so I will also try to explain them. Full title of the project is EVO-NANO platform for programmable nanoparticle-based cancer therapies, and since I guess you're not very familiar with nanoparticles and drug delivery systems, I will also explain briefly what they are and how they can be used in different fields. So yeah, the project is financed under the so-called FET Open call, and they define the target as support of the early stage research and innovation around new ideas toward radically new future technologies. And yeah, success rate now is 6%. A few years ago it was I think 1.5%. So it was absurd a little bit, and since competition is so high, we needed a few years to submit and resubmit and improve it, and then again resubmit. So after 5 iterations, we finally got it. Long-term vision of our project is here, is to create an integrated platform for the artificial evolution and validation of novel drug delivery systems for cancer treatment using nanoparticles. I will now try to explain each aspect of this, and we'll start with nanoparticles. They are, most generally, they are all particles whose size is between 1 and 100 nanometers. If something is smaller, it can be modeled and it behaves as atoms and molecules, but beyond 100 nanometers, you have behavior of so-called bulk chemistry or large-scale chemistry. So this scale between 1 and 100 nanometers is somewhere in between, and it usually demands new methods of both simulating and modeling their behavior, and also elucidating their properties. There is a bunch of them, and people are actually starting to investigate what can be used as a useful set of nanoparticles. Generally, they can be divided into organic nanoparticles and inorganic nanoparticles. Both of these groups have some pros and cons. With organic nanoparticles, good thing is that they can usually spontaneously dissolve in body, so their supposed toxicity is quite low, and they do not leave traces in organism. But on the other hand, with inorganic nanoparticles, we can have much better control of their size, behavior, shape, and pretty much everything. So you need to make a balance between what is actually your target. Again, their behavior generally depends not too much on underlying particle, but mostly on a manner how it is functionalized. By functionalization of nanoparticles, we mean attachment of additional chemical compounds, DNA molecules, etc., which will change their behavior. And yeah, I will go. Yeah, here's an example. So in this example, you have pretty much everything which is so far tested with nanoparticles. You can attach some drug molecules in native or crystal form. You can attach DNA or RNA sequences. You can attach ligands, receptors, whatever. Also, you can attach different dyes. So if you attach dyes, they can be much easily traced in organism, so they can be used for medical imaging or similar applications. What is important? Quite important is that you can produce so-called multifunctionalized nanoparticles. In other words, you can attach different chemical compounds on their surface, and in that way actually produce nanoparticles that can have different properties in different environments. Yeah, before I go further, I will go a step back with liposomes. For example, you can also pack. So, yeah, you can also pack whatever you want, basically, inside of them. So for example, if you want to deliver some drug molecules, you pack them inside of the micelles, and they are protected and do not interact with other parts of body except at the place of the delivery. So basically, the main strength of nanoparticles, of multifunctional nanoparticles, lies in the fact that in effect you can produce some form of simplified algorithms how you want them to react in the body or whenever you are applying them. So yeah, here is an example of organic nanoparticle which is multifunctionalized both on the outside and inside. As you can see, you can attach a so-called protective layer in order to protect it from so-called non-specific interactions in body. For example, if you inject them into bloodstream, they will immediately be attacked by either immune response or different proteins will attach to them and force them to coagulate. By different protective layers, you basically can cancel that effect. In order to target specific organs, you can add different proteins or ligands to their surface. So after some time in circulation, they will deliberately be attached to some specific type of cells or organs in body. After their attachment and increased concentration in some part of the body, you can engineer them to release their load. Release of load can be triggered by different factors. Slide, for example, one of the most popular techniques is to utilize slight variations in acidity in body. So if acidity increased for 0.2 degrees, they dissolve or they just open and then release their load. In this quite complicated example, they are loaded with both drug molecules and DNA string. why it can be used. For example, I think it was developed at MIT. They actually, by adding DNA string, you can provoke some response in a cell. You can activate, for example, some metabolic process, and then target it with a drug. So possibilities for manipulations are quite high. In standard chemistry, you have a bulk of same substance. You treat some organism or plant or whatever with it, and expect to have some effect. But here you can actually first target your delivery to specific parts of the host organism, but also you can modify metabolic state of the host organism and then attack some specific target you want. So possibilities of applications are quite high. Of course, since the field is quite new, there is a lot of additional research in possible toxicity of these compounds and some side effects. So, yeah, I already talked about this. The topic of my project is medicine, and but also there is some research, not many, just a few on, but also in other fields people recognize that with such possibilities for modification of host organisms, they can probably be applied to plants. And so far research showed that first, which is most important, you cannot go from each plant to each plant and inject it. So first line of research was to see whether you can apply nanoparticles on higher areas, and yeah, they showed that either through irrigation or through leaf surface, plants can absorb nanoparticles. Of course, it depends on the size of nanoparticles, but generally it is possible. Also, a few research showed that nanoparticles can transfer through both xylem and phloem and enter different parts of the plant, and of course can enter cells. And once when they enter a cell, the story is quite similar to medical treatment, because a lot of things are quite similar. But these main barriers, root and leaf system and xylem and phloem, are shown to be not so big problems as supposed previously. So back to this project. So why hybrid platform? The idea is to use as a first step artificial intelligence algorithms that will be attached to the model of circulation of nanoparticles, and this artificial intelligence should actually play against itself. It should automatically run some randomly initialized set of nanoparticles, evaluate its distribution and supposed toxicity, and then automatically go back at the beginning, tweak some of these properties, and then go again. So after several hundred rounds of playing against itself, we suppose that it will automatically optimize what kind of nanoparticles can be useful for a given scenario. In the next step, as a part of our consortium is a company which can synthesize these tailored nanoparticles, and finally we will test them both in vitro and in vivo using mouses and in vitro using microfluidic devices. Microfluidic devices are quite handy devices where I will be short about that. Where actually you can see actual size of them, but you can mimic a lot of aspects of real environment. So in standard in vitro procedures, you just mix some chemicals or mix chemicals with tissue culture and expect some results. Here you can usually they use nanolithography to make extremely small channels and chambers. Then at the walls, inside walls of these channels, you can add cells which can grow then inside. So you will basically reconstruct part of the organism which you need. But good thing is that once you do it, testing will be quite rapid, and also you will use a much smaller amount of chemicals in order to test their effect and their possible adverse effects. So yeah, this is the outline of the whole of the project's workflow. First, as I said, we will make a computational model, and during in silico testing, artificial intelligence algorithm will improve itself. In the next stage, we will construct 4 different microchips to mimic most important aspects of circulation through human body: vascular microchip, endothelial, tumor and cell area chip. And as a final stage, we will test them also on mice. Of course, these results from microchips and mice controls will be then fed back to the model in order to improve it. So during these 3 years, which will be project duration, we expect to complete at least 3 these full circles around the workflow. At the end, we expect first, if we were successful, I guess it would be a good step towards so-called personalized medicine, where you can rapidly develop a personalized treatment for each patient. Because here suggested composition of nanoparticles, their size, functionalization, and so on, will depend on a specific scenario. And it does not matter if you have some, I don't know, whatever kind of tumor, but equally important is where the tumor is, how vascular is it, what are the surface protein on it, and so on. So we plan to actually feed the model with all of these aspects and then produce nanoparticles for that specific case. And if it goes into streamlined procedure, then we hope that, well, not as a part of the project, but maybe later, we can make a good platform for each specific patient to develop a specific treatment for him or her. Yeah, thanks.